So this session is entitled Bacterium-host interactions, and we will have 4 speakers. The first speaker actually was supposed to be Ruth Ley, the director of the Max Planck Institute for Biology in Tübingen, Germany. But she got notification that just at the time when she's supposed to give her talk, she has to receive a Ukrainian refugee. So you see, the war in Europe has already impacts on Germany, and it probably will have much more. But we have a wonderful replacement for Ruth. There is Taichi Suzuki from Ruth's laboratory, and he's a senior postdoc since 2018 at the Max Planck Institute. Taichi did his studies on ecology and evolutionary biology at the University of Arizona and graduated from the University of California, Berkeley. His title today will be Co-Diversification of Gut Microbiota with Humans. So Taichi, the stage is all yours.
Thank you. Share the screen. Can you see my, yeah, I'll do the first. Yeah, can you see my screen now? And can you hear me well?
Yes.
Good, good. Yeah, so thanks for the introduction. And I apologize on behalf of Ruth that she couldn't join today. And I also heard that you're expecting a talk originally on lipids is what I heard, but I will not be talking about lipids. So I apologize on the context as well. So, but I hope you understand that we're supporting Ukraine, and she's acting as a leader. So thank you for having me, and I hope I can entertain you by sharing this story on the origins of human gut microbiome. So this work is already on bioRxiv, and this is a truly collaborative study that involved many people inside the lay lab and also outside the lay lab. So I'll be introducing them throughout this talk. And feel free to ask me questions and anytime during this talk. So as you may know, there are many, many microbes living inside and outside our body, and some can affect their health in positive and negative ways. But this is not new, right? We knew this for a long time, but what really changed in the past, say 15, 20 years, is this advancement in DNA sequencing technology. And now we can characterize the entire microbial composition and function through sequencing and these omics technology. So recent studies have shown these previously unknown roles of microbes, how they can affect our health in general, right? How they can affect our development of immunity, metabolism, physiology, all the way to behavior. But what the question I would like to ask today is pretty fundamental, is to ask, where do our gut microbiotas come from? So this is asking about the evolutionary origins of human gut microbiome. And there's 2 modes of transmission. So one is called vertical transmission, one is horizontal transmission. And I'm defining here pretty loosely. So vertical transmission is a transmission from parents to offspring. And horizontal transmission is a transmission from other individuals or from the environment. So at least in mammals, we know that we're born mostly sterile, and we get the first microbes from our mother's vaginal canal and their skin and breast milk and so on. And we also acquire microbes through diet from other individuals later in life. And the community gets more and more complex, and it gets pretty stable during adulthood. And this process of community assembly is pretty similar to any ecosystem like the forest. And we also know there's important priority effects on the microbiome community as well. So these initial colonizers of the these microbes are important to determine these community composition and function later in life. So it's important to understand, where do we get our microbes in the first place, to understand how the microbes affect our biology. So I've studied these transmission modes of bacteria. So this is the work with Andrew Muller back at UC Berkeley, that we collected these wild house mice from nature, and we do brother-sister matings for more than 10 generations over 3 years, in this case from 2 populations. And using this data set, we studied these transmission modes of bacteria. And here's the main finding from this work. So if you take this transmission score as the x-axis, you can see this distribution is not normal, right? So you can see there's a significant bimodal distribution. So there are microbes that specializing, seems to specialize more on vertical transmission, which tend to be anaerobes, which cannot live under oxygen. And there's some seems to specialize in horizontal transmission, which tend to be aerobes. And these microbes often associate with these hospital-associated microbes. So this finding was interesting, that there seems to be microbes that's anaerobic and transmits across multiple generations. So the obvious next question is, does this happen in humans? Oops. So the main question of this work is to ask if there are microbes that have been vertically transmitting for thousands of generations in humans. So this is a map showing the ancient dispersal patterns of modern humans in the past 100,000 years. So as you know, we originated in Africa and spread across the world and formed these genetically distinct populations. So if there really are microbes that's transmitting from parents to offspring for thousands of generations, we might expect some microbes to show a similar migration pattern as humans. And the most famous work is Helicobacter pylori strains, which match the patterns of human migration. So these microbes mostly live in the upper gastrointestinal tract, and people have isolated these bacteria. And if you genotype them, you can see the upper panel, it's showing you the nucleotide diversity of H. pylori. And the bottom panel is showing you the inferred migration history of the bacteria. So you can see it's consistent with our migration history. So this work is pretty impressive. And we were wondering, besides H. pylori, can we identify other gut microbes that co-diversify with humans? So one approach is to test for co-phylogeny, which is to compare host phylogeny and microbial phylogeny and look for congruence. So for example, if you look at the very left panel, if you see this matching pattern, it's a pattern called co-phylogeny or co-diversification, and it's consistent with vertical transmission. And on the opposite end, on the extreme end, when you see these mixed mismatch pattern, it suggests there's a frequent exchange of these strains between these host populations. So it's consistent with horizontal transmission. So the idea here is pretty simple, that among these thousands of species of gut microbes in the human gut, can we rank them by their degree of co-phylogeny? And people have done similar work. So Nick Youngblood in our lab have shown how mammals show signs of co-diversification with gut bacteria. So he looked at different vertebrates, so fish, amphibians, birds, mammals, and reptiles. And you can see the mammals show the strongest signal of co-diversification, especially herbivores. And perhaps this is due to some mammal-specific traits like milk production. So this work was done using a single gene called 16S ribosomal gene. So these gene fragments are short, so the taxonomic resolution is limited. So Andrew Muller did this work within mammals. These hominids show signs of co-diversification as well. So on the left, we have the host phylogeny. On the right, we have the bacterial phylogeny representing this family Bifidobacteriaceae, which includes this genus Bifidobacterium, which is famous for mother-child strain sharing. So you can see this congruence except this gorilla branch. So this overall congruence suggests that these bacteria may have been is sharing this evolutionary history over millions of years of evolution. So this is also done using a single gene called gyrase B. So this gene is longer, so you have better taxonomic resolution, but the number of bacteria families that we can survey is limited. So the next obvious question is to ask if this co-phylogeny, co-diversification signal extends within humans, and this has not been done. But at least we know that these human populations have different strains of specific gut bacteria. So more of these recent methods like StrainFlan uses these multiple marker genes from metagenomes and try to study these strain diversity. So for example, this is showing you the global strain diversity of Bifidobacterium longum. And you can see each country is dominated by different microbial strains. But to really test for co-phylogeny, we need matching sets of human genomes and metagenomes. And this has been lacking in the field. So as a lab, we went out and collected these paired samples from 3 countries, Germany, Gabon, and Vietnam. So we're mostly recruiting mother and their child. And through collaboration, so this work was led by Victor, Liam, and Nirbu in the lay lab. And through collaboration, we also added populations from the United Kingdom and Cameroon. So at the end, we have this paired data set of human genomes and fecal metagenomes from 747 adults and also 386 children for the metagenomes. So we'll be using this data to explore this question. And here's some results. So this is showing with the human genetic relatedness represented as a phylogenetic tree from our data set. And you can see the expected pattern. So individuals from Asia and individuals from Europe form a monophyletic clade, which are sister to each other, which are nested within individuals from Africa, which is exactly what we expect. And I also like to note that we also collected subjects from hunter-gatherer populations in Cameroon and Gabon. And these populations live in a geographically separate location, but they're each other's closest relatives. And I'll come back to this in a second. So this is the human phylogeny. So now let's look at the microbial phylogeny. So here are some top lines. You can see some microbial trees look really similar to the human tree. So for example, Roseberia hominis and Collinsella aerofaciens, you can see the most basal branch are strains from Africa, and strains from Europe and Asia are more derived, which is consistent with our tree. And when you look at Prevotella copri or Eubacterium rectale, the tree looks more complex. But when you look at each subclade, you can see a consistent pattern. So the most basal strains tend to be African strains, and Asian strains and European strains tend to be more derived. So this suggests that perhaps there's multiple transfer events from Africa to the rest of the populations. So coming back to that hunter-gatherer population, so despite their living in a separate geographic location, their strains are also each other's closest relatives, suggesting some roles of vertical transmission and argues against these acquisition of microbes from the soil, for example. So these are the top taxa, but when you look at most of the other taxa, they look like these. The colors are mixed and there's not much structure, like these Parabacteroides and Bacteroides species. So this suggests that these strains have been transmitting between populations frequently throughout this history. So we can keep staring at these trees, but we can also run statistics. And we try to rank the microbial taxa based on the evidence of co-diversification. So we applied 3 different co-phylogeny tests on 2 different types of trees. So let me explain this a bit. So these co-phylogeny tests are permutation-based tests that comparing 2 different trees, distance between 2 trees. So the observed tree and the permutated tree. So that's how we're getting the p-values. And we're applying this to 2 different types of trees. So best maximum likelihood tree and consensus tree, meaning we're collapsing the branches with low bootstrap support. So at the end, we ran 6 different tests. And when all 6 were significant, we gave a co-phylogeny index of 6. When nothing was significant, we gave a co-phylogeny index of 0. So we can rank this in many different ways, but we get overall consistent result. So when you do this, you see some interesting patterns. So for example, bacterial species that belong to the phylum Firmicutes, shown in blue, show greater evidence of co-phylogeny compared to species that belong to Bacteroidetes phylum, which is shown in red. So there's some taxonomic signal. And this also makes ecological sense because these taxa that are pandemic, that you can found in many populations, are used as markers for fecal pollutions in rivers. So there are experimental evidence showing that these microbes can survive and be dormant in water for certain periods of time. So this can be a mechanism that they can spread across different populations. And this finding was pretty surprising, that top taxa, so that microbial species that show greater evidence of co-phylogeny show smaller genome size. And this is very exciting because when you look at the insect symbiont literature, this degree of intimate symbiosis is associated with a smaller genome size. So this suggests some interactions between mammalian host and the microbes. And we were first worried that if this relationship was driven by the phylum genome size difference. So when you look at within each genus, you can see 7 out of the 8 genus showed this consistent slope. So there's some signal to this within genus as well. And we're currently revising this paper, and we're looking at the GC content, and you can see this trend of AT-rich genomes in more co-phylogenetic taxa, which is also consistent with the insect symbiont literature. And also, Kelsey has a postdoc in the lab that experimental work showing how microbes that show greater evidence of co-phylogeny are more sensitive to oxygen. So this is consistent with what I found in mice, that taxa that transmit vertically tend to be anaerobes. So this suggests there's some adaptation to the host environment. We also tested if this co-phylogeny signal extends within population. So what I mean by this is so for example, if you make these trees only using African samples, you can see the signal extending within Africa. And this tend to be the top taxa. And I see this as a support of vertical transmission because say if there's a dietary factor that's driving this co-phylogeny signal using all samples by chance, and we don't expect the same dietary factor to drive these co-phylogeny patterns within populations. So this argues against these environmental acquisition for at least these top taxa. And we also trying to quantify these strain transfer events using stochastic character mapping, and this is consistent with the out of Africa migration pattern. So what I'm showing you here is that each dot is a microbial taxa, x-axis is co-phylogeny index, y-axis is the proportion of transfer events, transmission events from Africa to other populations. So for example, if you look at these top taxa like Collinsella, you can see the ancestral state is African strain, and you can see the African strain may have transferred to Asian population and European population. So that's why you have a higher proportion of transfer events from Africa to the rest. And you don't see this for taxa with local phylogeny index. So we can do this for many, for other major human groups. So you can see these transfer events from Asia to other population do not predict the degree of co-phylogeny, but transfer events from Europe to other populations do predict this co-phylogeny index, but in the opposite pattern of that of Africa to other population. So what I mean by this is that these taxa that show no evidence of co-phylogeny, meaning these pandemic strains, tend to have European origin. So this is consistent with the colonial history that these microbes these pandemic strains can be coming from Europeans to the rest of the population. So overall, this is consistent with our migration history. So what I showed you so far is the result based on 5 populations. So how general are our findings? So to get to this, we added public metagenomes and see if the results are consistent. So we added more than 1,000 human fecal metagenomes from 19 countries, and also more than 100 primate metagenomes from 27 species from natural populations. So these are all public data. And we compared these strain transferring events between top 10 and bottom 10 taxa. So here's some examples from the top 10 taxa after adding the public metagenomes. So first I'll orient you. These inner branches are corresponding to more coarse genetic structure, and these outer circles are corresponding to more fine genetic structure. So first thing you can see is that these colors tend to cluster to each other, so suggesting that there's a population-specific strains. So these signals that we detected with 5 populations is pretty robust. And when you look at these strains from primates, they tend to be the most basal strain, followed by African strains. And these Asian strains and European strains tend to be more derived. So again, this is consistent with our evolutionary history. So this is the top 10 taxa, but now let's look at the bottom 10 taxa. So you can see the patterns are again random and you don't see much structure. And these primate and African strains are all over the tree. So this suggests that these strains has been transmitting between populations and even between species, between humans and primates throughout this history. So when you run the stochastic character mapping, I'll show you that next. So when we run this, so this is the result I already showed you, but in a different form. So I'm comparing the transferring events from Africa to the rest, comparing the top 10 and bottom 10 using the initial 5 population. And you can see the top 10 taxa tend to have African origin. And this is after adding the public metagenomes. You see a consistent pattern where these top 10 taxa tend to have African origin compared to the bottom 10. And this is, I haven't shown you yet, but when you run this for child metagenomes, you can see a similar trend. So there's one outlier here. If you exclude this, it's significant, but you can see the trend. The top 10 taxa tend to have African origin. So now let's look at the child samples. So the list, the co-phylogeny list for child is dominated by Bifidobacterium species. So you can see it's only from 3 populations and sample size is small, but you can see there's no clear basal group, but again, it clusters by population. And when you look at the ranking, all 4 Bifidobacterium species that we tested were significant. And this also extended within population as well. So again, this is very exciting because Bifidobacterium is famous for being shared between mother and child. And you might be wondering that these taxa that we found in adults for co-phylogeny may show up in the child list as well. But this was very challenging because the composition between adults and child is very different, and we need enough coverage to make these phylogenies. So we couldn't test the same species between adult and child for many of the taxa. But despite this power issue, we found that Prevotera copri showed evidence of co-phylogeny in both adults and children. So again, this further supports this role of vertical transmission. But this is still indirect. So we also try to test this in a more direct way by looking at mother-child strain sharing. So this work was done by Hagai and Liam. So Hagai actually developed this tool called SynTracker. So this is using long stretches of DNA and look for synteny between 2 genomes. So that's how he's calculating this strain similarity score. And Liam used this software called InStrain developed by Joe Banfield's lab that using a population level nucleotide identity to look for strain relatedness. So when you do this, they're both using different methods, but they came up with a similar list of species. And this list was dominated by Prevotella. So how to read this is these blue bars are the strain similarity between related mother-child pairs, and these gray bars are strain similarity between unrelated mother-child pairs. So you can see these top taxa, these strains are more similar between related mother-child pairs. So now let's think about the mechanisms that can be driving this co-phylogeny. And these drivers of co-phylogeny is likely not only mother-child strain sharing, but also within village transmission. So let me explain this. So again, these blue bars are strain transferring events between related mother-child pairs. The gray bars and gray numbers are strain sharing events between unrelated mother-child pairs. So when you first focus on Gabon, you can see the majority of the strain sharing events are happening between mother and their own kid, right? But we also detected these strain sharing events between mother and unrelated child living in the same town. But in case of Gabon, this happens even between towns as well. And you can see similar pattern in Vietnam, but the pattern was very different in Germany. So we didn't detect even a single strain sharing events between mother and unrelated child in the same town. Even we compared many mother-child pairs. So this difference between potentially Western and non-Western countries, we don't know the mechanism driving this pattern. It can be cultural differences, sanitation differences, hospital differences. But the main message that I would like you to take from here is that this mechanism of co-phylogeny is not only restricted to this strict mother-child transmission, but also this village-level transmission that the people that are living in close proximity. So just to summarize this work, so we provided a list of gut microbial taxa with evidence of co-diversification, both between population, also at the within population level. And we supported this result by adding additional populations and looking at mother-child strain sharing. So overall, this result support this role of long-term multi-generational vertical transmission, explaining at least some of these patterns. So I would like to close this talk by just to think about the roles of these co-diversified strains in human health and evolution. So why this study matters. So here's a map showing you the classic examples of local adaptation in humans. So human geneticists have identified these adaptive loci that's involved in adaptation to different climates, different diet, different pathogens. And all these famous examples, there are associations with microbial composition and function. So many of these taxa can perform a similar function as the adaptive allele, like producing similar enzymes, for example. And a lot of these bacteria show evidence of co-phylogeny. Some are vertically transmitted, and these bolded ones even have genetic basis, meaning that these adaptive alleles can predict the relative abundance of the bacteria that performs a similar function as the adaptive allele. So these examples, I think it's exciting that these co-diversified strains or these population-specific strains can contribute to local adaptation, like speeding up or slowing down the process of adaptation, and it can involve in local health benefits, like the concept of personalized medicine. So this work can have implications to basic ecology and evolution, but also to biomedical research. So for this, I'd like to thank all the people who are involved in this study. And thanks again for listening for this different topic than we expected. So thanks for listening, and I'm happy to take any questions. Oops. So I see a hand, but how should we?
Can I ask you?
Yeah, please.
Very interesting. I enjoy your talk.
Thank you.
My question is, given the increased usage of cesarea for birth, is that going to? Do you expect that would change the? I mean, what is the main mechanism of transmission of the vertical transmission?
No, that's a great question. So you meaning that C-section procedure? Yeah. So yes, we know that the vaginal birth babies and C-section babies do have different microbiomes, when they're born, and there are mixed results now that later in life, it can come back or it can stay different, and there's some mixed studies. But yeah, that's a great question, and I think to be honest, this our study alone cannot really tell, when these microbes are transmitting from mother to child, right? It can be through the vaginal canal, it can be these nursing stages, or it can be later in life, just sharing the same habitat, shared environment alone, eating the same diet. Maybe it's a cultural thing, right? If you eat the same food, similar food. So we don't know the mechanism, and that's a great question that we have to dissect. And as a field, it's really hard to show this vertical transmission versus shared environment. So that's a great question, and that's something we have to dissect, and we don't have a clear answer. Yeah, great point.
Mauro.
Hello, Taichi, can you hear me?
Yes, yeah? I can hear you.
Well, very nice talk, and I'm going to make a question by observing some of your data, which have a lot of genomes, a lot of statistics. Because I'm an expert of proteobacteria, I noticed that in the list of the co-phylogeny you started, there is only one, which is Escherichia coli, essentially. However, when you move to the children microbiome, I see there is also Azospirillum, which is presumably are the Azospirillum MIGs, which have very anaerobic, which I've actually been working on. We have been working on those. And from the brief analysis you presented, it looks like this particular Azospirillum doesn't fit any co-phylogeny. Therefore, my question is, does it mean that this Azospirillum and maybe other alpha proteobacteria, which you can find more obviously in the children microbiome, are just coming from the environment, from the diet?
No, that's a great question. And let me explain how we selected these microbes, because it's not comprehensive in a way. So we selected very prevalent bacteria, so bacteria that exist in all populations for a certain number of individuals. So we're only testing microbes microbial species that's present in all these populations above certain relative abundance, per se. So that's already selecting a lot of population-specific microbes that we could have tested, but just based on the power, we only looked at very common bacteria. So that's why it's biased that there's less proteobacteria. So the simple answer is, I don't think we tested proteobacteria in a comprehensive way. So I don't think we can say anything about them. And your comment of about anaerobic proteobacteria, that's interesting, because a lot of proteobacteria, as I understand, is more facultative, or some are aerobic, right? So I think it'll be great to study proteobacteria focused analysis than what I showed, because just the sample size, how we selected, I don't think I can make any statement to your question. So that's a great question to follow up. Yeah, thanks.
Esperanza.
Hi, Taichi. Wonderful talk.
Thank you.
I enjoyed it very much. Did you present a hypothesis where you say that those that are conserved maybe have an adaptive role as humans colonize different places? No, your conclusion.
Yeah.
I was thinking of an alternative explanation, because if they are conserved in the humans and they are vertically transmitted, then maybe they have a more essential role independent of adaptation. And those locally acquired bacteria would have more a role in local adaptation to novel food, to novel things, and then these are more essential for digestion or for immune responses. And the other question I had was on sporulation, because many sporulating bacteria seem to be acquired from the environment. So does that correlate as well?
Yeah, great question. For the first point, yeah, that's something I think about a lot. And, just the simple answer is it can be mixed, right? It can be just no function to the human at all. It can be just a bicarience event, just hitchhiking the human population. There's not much, it's just maybe a transient bacteria that, is a possibility. And what you suggested is an interesting one. So if it's really fundamental to our biology, if we need these bacteria, we expect there's no variation, right, of those essential bacterial genes, for example. But given that we are seeing these strain variation by different population, and there's some studies showing these carbohydrate metabolism for Prevotella and Eubacterium is population specific. So this is something that what you're thinking, it can be, they're adapting to the diet of the local population. So this is all possible. And the second point of the spore forming, we haven't really tested explicitly, so we don't know, but I expect spore forming bacteria will be more, like you said, more show less evidence of co-phylogeny is what I expect, as you said. So so those are all great questions.
Okay. Thank you. First of all, thank you for this splendid talk. I had a very general question. It's in relation to the Esperanza question also, is what do you consider is influence more in the microbiome of each individual, the race, the sex, or the environment?
I see. So you're asking the selection pressure on the bacteria.
Okay.
No. I'm trying to clarify the question. So you're asking what type of selection pressure?
No. Yeah, my question is specifically what do you think influence more in the microbiome of each individual, the sex, the race of the environment? I see.
So I think if you think of it, microbiome as a community,
yeah.
Yes, diet can affect, host genetics can affect, and based on how you set up the study, these proportion, contribution can change. But what I'm focusing here is a strain identity. It's more about lineages of bacteria. So the idea is, whatever you eat, maybe the abundance will be low, but these strains can be present in low abundance. And when you have a child, maybe you transfer, transmit these bacteria. So maybe it doesn't affect it by environment, for example. It is possible for these strain identity wise. But in terms of composition, if that's your question, yes, it can be affected by all sorts of factors. So.
Thank you.
Thanks.
So thank you very much, Taichi, for a wonderful presentation. And you saw there was a vivid discussion on your results. So let's hope we can interact in the future. I heard you got a position in the US for a professorship.
Yes, still it's under negotiation, but yes, I'll be closer to you guys.
Okay.
So, yes, I'll be happy to interact in the future for sure.
And come south to the border. Yes. Okay, great. Thank you very much.
Yeah, so, thanks for having me. Yeah.
So my name is Otto Geiger. I think you know me, so I don't need to give much introduction on that. I'm going to talk today about recruiting fatty acids for bacterial sphingolipid biosynthesis. Now, let me shortly outline what is the belief, the model for a cell envelope in gram-negative bacteria. And essentially, we can distinguish the inner membrane with a phospholipid bilayer, which is surrounding the cytoplasm, the outer membrane, which on the inner layer, it really has phospholipids, and on the outer layer, it is thought to have lipopolysaccharides. Between the two membranes, there is the periplasmic space filled with the cell wall, and these are essentially the ideas from beginning of the 90s, here outlined by Ritz and Doden. Now, when German and Japanese researchers studied one bacterium, which was called Pseudomonas paucimobilis in more detail, they found out that this Pseudomonas paucimobilis has no lipopolysaccharides. Instead, they found sphingolipids. Now, this was such a surprise that the bacterium was renamed. It was now called Sphingomonas paucimobilis. And every bacterium that now had sphingolipids was called Sphingo something. Sphingomonas, Sphingobacterium, Novosphingobium. But a few kept their names, like Sorangium cellulosum, for example, no? Which we see here. And the idea here is that in some bacteria, sphingolipids might replace partially or completely lipopolysaccharides in their function. While sphingolipids got their name from their discoverer because They were so enigmatic as the Great Sphinx of Giza in Egypt. And you can see here a sphingolipid, a relatively simple one from a bacterium, Sphingomonas wittichi, which has a long chain base here, substituted, amidified by another fatty acid residue, and a small head group of an uronic acid. The biosynthesis of sphingolipids in eukaryotes is pretty well known, especially from yeast and animals. And what we have here are several steps. Step one, where a serine palmitoyl transferase condenses serine with palmitoyl coenzyme A. And this is a decarboxylative condensation forming 3-oxosphingony. So this is the first step. The second step is a reduction of this keto group to a hydroxy group by a keto-sphingonin reductase. And that is forming the long chain base sphingony. In the third step, a ceramide synthase is putting another fatty acid on this long chain base forming dehydroceramide. And in a fourth step, in eukaryotes at least, there is often a unsaturation introduced into the alkyl chain forming the ceramide. Then there would be different reactions to put head groups on this lipid anchor. Now, what about bacteria? The problem there is that it's sometimes difficult to predict which bacteria might make sphingolipids, because if one is searching with proteins for homologues in bacterial genomes, well, one only finds homologues for this serine palmitoyl transferase. And what you see here is a tree of bacterial alpha-oxoamine synthases, how they're called. And there are several enzymatic activities sorted together. One which is a ligase really, another involved in heme synthesis, and then this BOF, which is involved in biotin synthesis, is very frequently actually assigned for serine palmitoyl transferases in bacteria. So sometimes you find 2 BioFs assigned, and you have to look at genomic context and see that this is probably not correct. So there are serine palmitoyl transferases from eukarya and from bacteria, and there are 2 subgroups. One subgroup is really in those Bacteroidetes, which we have now heard in the previous talk, and they're also called Cytophaga flexibacteroides group. And you see them here. And there is another group which is formed by representatives from the alpha, beta, and gamma proteobacteria, which are combined as rhodobacteria. Now, remarkably, the serine palmitoyl transferases in rhodobacteria, the structural gene for that is preceded by something that looks like a gene for an acyl karyoprotein. And that got our interest. So is this really an acyl karyoprotein? Is there another mechanism going on? Our model for this study is Caulobacter crescentis, which got some fame because it's really a model for asymmetric division. And essentially from a sessile cell, there is an asymmetric division to form a flagellated moving cell that then can form later on a stalk again and become sessile and initiate a new cycle. We chose this bacterium because it's well studied, it has good genetic tools, and there's relatively much information. Okay, here you see one of the results, which is more an example of a result where we separate lipid extracts in thin layer chromatography. And here, for example, you see the standard ceramide, the one from Sigma. And you can also see that in those extracts of wild-type Caulobacter, there are at least 2 compounds formed which migrate similarly, which might mean they are ceramides or dihydroceramides. These are resistant to treatment to alkali, so they are not typical phospholipids because they would not be resistant to treatment to alkali. However, the ceramide would be. And you also can see that in a mutant deficient of serine palmitoyltransferase, these signals are not appearing. However, if we complement in trans, they're appearing again, which is not the case with the empty vector. So essentially with all the mutants I will show you now very shortly, we did these kind of experiments, these genetic experiments, make mutants, complement them. So we have the typical loss of function, gain of function experiments that would then confirm the involvement of individual genes. Now, where is serine palmitoyltransferase located? You see here a cluster of genes that is organized in 6 operons. And just down here, you see the serine palmitoyltransferase. Now, what is interesting is that there are several studies, genome-wide, one from Kristen that defines the fitness for each gene in the colobacterial genome. And as you can see, all those with pluses, if there are mutants in those genes, they would be affected in fitness. And that actually has been assigned as an essential gene, which is probably not correct. Now, there is another more recent study where with all those mutants, they made now hundreds of experiments of growth curves in different media to define something that is co-fitness. So which mutant would show co-fitness with serine palmitoyltransferase, which is most similar in their phenotypes with the mutant in serine palmitoyltransferase. And if we have a number of 1, this would be a perfect match. But you see we have numbers of 0.92 and so on, but they're all pretty high here. No? There's another one high down there. So this means mutants in those genes would behave similar to mutants in the serine palmitoyltransferase. And it also means there is a good chance they're involved in the same biochemical pathway. Okay, so I now will shortly focus on those genes, the predicted acyl coenzyme A synthase, predicted epimerase, which might be the key to sphingolipid reductase. This acyl carrier protein, which I told you before, is upstream of serine palmitoyltransferase, and another one, which is a potential N-acyl transferase, and which might make the last step before forming dihydroceramide. I will not have the time to show you all the data, and it's probably not necessary because this has recently been published. And if you are interested in the detail, the reference is down here. But these mutants deficient in serine palmitoyltransferase, what I could show you is that they do not form ceramide. That was the thin layer chromatography. That they do not survive in stationary phase at elevated temperature, and it's really a dramatic effect. That they can grow in the presence of polymyxin B, whereas the wild type cannot. That they're sensitive to detergents, suggesting they're affected somehow in their membrane properties. Now, the mutants in the 4 other genes, 11, 54, 65, 64, and 63, they show all of the above mentioned phenotypes. So this explains this high co-fitness, for example, or at least with those phenotypes we studied. And as we suggest in this paper, these 5 genes, so serine palmitoyltransferase and the other 4, are involved in forming ceramide in bacteria. And this is the proposal. So there would be an alteration in the first step, especially of sphingolipid biosynthesis in bacteria, where a special acyl carrier protein is loaded with 3 fatty acids to form an acyl ACP derivative, a palmitoyl ACP derivative, and this is catalyzed by a special acyl ACP synthase. In second step, the serine palmitoyltransferase would then preferentially use this ACP thioester derivative to condense it to serine and form 3-oxosphingolene. And then there would be the usual steps of a reductase and of an N-acyl transferase to form the dihydroceramide or ceramide. I'd like to focus now on this upper part here to see whether we can verify this to some extent biochemically. And what you can see here is an acyl carrier protein, a representation of an acyl carrier protein and its prosthetic group 4' phosphopentethine. Now, one building block of 4' phosphopentethine is beta-alanine, and you can use actually beta-alanine, labeled beta-alanine, to see whether it's getting incorporated into this protein or attached to this protein. Now, here are cell-free extracts separated in a 20% native page. And the protein we're talking about is this CC1163, this special acyl carrier protein. You see it's expressed as a nice band. We have a similar protein from Escherichia coli B. And you also can see that this gets labeled with beta-alanine in a parallel experiment where we simply offer this radio label. So this radio label suggests that really there is a phosphopentethine attached, and this has been confirmed by mass spectrometry. I will not have time to show you the data. If we now use this special acyl carrier protein with its phosphopentethine residue as a substrate, and as another substrate, radio-labeled palmitate, then we see incorporation of this palmitate into that protein, in this special acyl carrier protein. And you see this slight increase here over time. So this is really the kinetic of that reaction, whereas this does not happen if we offer palmitoyl-CoA as a substrate or palmitoyl-ACPP, which is the household acyl carrier protein used for fatty acid biosynthesis, for example. So we can conclude from that that in rhodobacteria, AASR, this special acyl ACP synthase, selectively acylates this ACPR, this special acyl carrier protein. We also have checked that with another method, and what you clearly can see here is that for getting a protein labeled with palmitate, and we separated then that in the thin layer chromatography, in the polyacrylamide gel electrophoresis, made an autoradiogram, and only when we have the special acyl ACP synthase, this special ACP gets labeled. In a control experiment, if we have the constitutive acyl carrier protein, well, only acyl ACP synthase from Vibrio Harvey can make the label on this protein. So there is specificity going on, and it's covalent linkage. Now, this is the experiment that shows how serine palmitoyl transferase is acting and what it has as preferred substrates. So, for example, you can see very clearly if we use palmitoyl ACPR, so this specialized acyl carrier protein, and a cell-free extract in which different serine palmitoyl transferases had been expressed from Caulobacter, from E. coli B, or from Cingomonas wittichi. In all three cases, the thick and fat band formed that migrates like 3-oxosphingolipid. So this would be relatively good substrates. This, however, is not the case if we use palmitoyl coenzyme A, which would be the typical substrate in eukaryotes. There is a slight formation of this band, but it's much less. And there is also a very slight formation when we use the constitutive acyl carrier protein. So clearly there is a preference of bacterial serine palmitoyl transferases, at least from the proteobacteria, for this special ACPR. So this confirms actually what I showed to you before, that free fatty acid is condensed with a special ACP catalyzed by a special ACP synthase. And then we have this loaded ACP that is then condensed with serine to form the 3-oxosphingolipid. Now, this is a little bit a crazy scheme in the sense, free fatty acids. There should not be much free fatty acids in a cell or in an organism. So where do they come from? Because free fatty acids, as they're sold, they would be pretty disruptive. They would act as detergents. So they cannot be there in high concentration. So they must come from somewhere. And studies of Isabel Lopez Lara and students of her will really suggest that in addition to biosynthesis of membrane phospholipids, there is turnover of those phospholipids, so degradation. This is like a steady state situation where you get permanent liberation of free fatty acids. In order to support this idea a little bit, we now tested whether in those different mutants there would be at least transient accumulation of fatty acids. And that you see here, that in the wild type, well, there seems to be a transient accumulation of compounds migrating like fatty acids, well, 7 hours after we had an optical density of 0.1. In a mutant deficient in SPT, well, this seems to be extended and even goes into the stationary phase, this accumulation of fatty acids, suggesting there is less use of fatty acids for sphingolipid biosynthesis. And this is also the case for the ACP-R mutant or for the AAS-R mutant, and to a lesser extent for the dehydrogenase and the N-acyltransferase mutant. So let me summarize. This is not the summary of my talk. This is more the summary of the last month of history. And, but let me replace Uncle Sam by Caulobacter crescentus. And Caulobacter crescentus tells us, I want fatty acids for sphingolipid biosynthesis. And the nearest recruiting station for that is ACP-R, AAS-R. So I'm finishing showing the collaborators in that project. So thanks to all previous collaborators and all the people listed here. I want to emphasize especially the two Jonathans who did most of the practical work for receiving, well, their PhD thesis and their publications, Jonathan Padilla and Jonathan Olea, two wonderful students who did an extraordinary job. Thank you very much. So maybe, are there any questions? Mauro?
Fantastic presentation. I know most of it, but would you elaborate briefly the importance of this work in the context of a eukaryogenesis, evolution of our cells?
Well, that would be a lot of speculation, really, because, well, let me point out the difficulties first. The difficulties are that sphingolipids are typically eukaryotic, or are considered typically eukaryotic, and that they are formed, well, especially in the endoplasmic reticulum, and then some ongoing reactions like modifications in the Golgi, and then they're transported to the plasma, to the surface of the plasma membrane, decorating the outer surface of the plasma membrane. So now one has to get this somehow together with a possible bacterium that integrated, let's say, some alpha proteobacterium, no, if you want the mitochondria, but then it should have migrated some of the biosynthesis parts to other organelles. Well, the connection between endoplasmic reticulum and mitochondria is not so far-fetched. So this is imaginable, not that some of the biosynthesis apparatus migrated from a possible alpha proteobacterium to some of those organelles. But one needs to make several speculations if one wants to explain that eukaryotic membranes somehow resemble bacterial membranes and not archaeal membranes, although eukaryotes are thought to be derived from archaea.
Otto, thank you. Perhaps this is a rather simpler one. Could you expand on the role of sphingolipids? I was curious about this phenotype that you had, where in the absence of one of the parts of the biosynthesis route, you had resistance to polymyxin B, right? Are you suspecting some charge differences? Because they were also susceptible to detergents, right? So.
No, so usually the target of polymyxin is thought to be lipopolysaccharide, but lipopolysaccharide that has two phosphate groups at the different ends of the molecule. Now, there are some bacteria, especially alpha proteobacteria, which do not have this modification. So they're probably not sensitive to polymyxin based on the lipopolysaccharide structure, but they're based on the sphingolipid structure. And there are indications that there is a phosphosphingolipid. They are sensitive. So we also have some other mutants which are involved in the decoration of the ceramide, where they cannot make phosphosphingolipids anymore, and they would be resistant to polymyxin B. So the mechanism in Colobacter is different in the sense that there is a different target molecule for adhering for polymyxin B.
Great, thanks.
So I think we need to move over to the next talk. So let me get my act together.
All right. Share.
Okay, so the next talk will be given by Jean-Michel Ané. And Jean-Michel, we have you already here. Great. Wonderful that you're with us. And Jean-Michel really originates from France. He did his undergraduate studies at the University of Lyon, then moved to Toulouse, where he did his PhD studies with Jean Denarier and Charles Rosenberg. And then finally he moved to the US, first to the UC Davis for a postdoc, and afterwards as a professor to the University of Madison, Wisconsin. And there he moved up the ranks. So for several years now, he's full professor at this university. His interests are really in plant microbe interactions, in nitrogen fixation, in symbiosis, but also extending it to cereals, to non-legumes, these kind of interactions. And I think this is what he will be talking today. Yep. So glad to have you here. Please go ahead.
Sure. Can you hear me well? Okay. All right. So thanks a lot for your invitation and the opportunity to talk about nitrogen fixation in cereals, one of my favorite topics. And in particular, today I'm going to talk about how we can improve the benefits of nitrogen fixation through what we call associative nitrogen fixation. You had many talks on rhizobia associations, but today I'm going to insist on associations that do not lead to the formation of nodules, but rather when the bacteria are more on the surface of different plant tissues. So I'm going to start a bit broad, and I know that many of you know about nitrogen fixation already, but I'm going to start with the nitrogen cycle, just to remind you that most plants, including cereals in particular, take their nitrogen mostly from the soil in the form of nitrate and ammonium. What we call nitrogen fixation is the conversion of dinitrogen from the air into ammonium, a form that the plants can take up. Industrial fixation is also called the Haber-Bosch process, was invented at the beginning of the 20th century, and is the conversion again of dinitrogen into ammonium. but using fossil fuel, using natural gas as energy source. And today, industrial fixation, the synthetic fertilizers are, as you know, a major source of nitrogen in our cropping systems. I don't know if you know that you have nitrogen fixation happening when you have lightnings. It's what we call atmospheric fixation. But today I'm going to focus mostly on biological nitrogen fixation, which is performed by bacteria and archaea that have what we call the nitrogenase complex. And some of our crops, like legumes in particular, are really good at associating with some of these nitrogen-fixing bacteria. And that's why we have legumes in all of our cropping systems around the world. And these plants, legume crops, they don't need a lot of nitrogen inputs, it's sometimes nothing. And they even can add nitrogen to the cropping system through crop rotations. But unfortunately, when it comes to cereals, most of our cereal crops don't benefit a lot from nitrogen fixation. So that's the problem that I want to talk about today. To put numbers on the different types of fixation that I just talked about, atmospheric fixation through lightning, it's rather minor. It's less than 10 million tons of nitrogen per year. Biological nitrogen fixation on land, so excluding the oceans, estimates vary between 90 to 150 million tons per year. And industrial fixation, again, something invented fairly recently at the beginning of the 20th century. According to the last estimate I was able to find last year, it's 170 million tons per year. So even exceeding biological nitrogen fixation at this point. So right away, you can see that it's going to cause a major disruption of the nitrogen cycle. So nitrogen fixed through the Haber-Bosch process, first, we need to acknowledge is a good thing because it's literally feeding half of the world. And here, so in that study, here it's a simplified graph taken from that paper in Nature Geosciences. They looked at the growth of the world population, here in red, that's the increase of the world population. And here in blue, it represents the proportion of that population which is fed through synthetic fertilizers. So the Haber-Bosch process. And most estimates say that today about half of the world population, if not more, is sustained through these synthetic fertilizers. So obviously it's a good thing, and nobody would propose today to get rid of that, of course. We heavily rely, especially since the Green Revolution, on the use of synthetic fertilizers. But as you know, there are also some negative consequences. If you look at the big picture, I think the most concerning aspect is sustainability. Because the Haber-Bosch process, like any mechanism for nitrogen fixation, requires a lot of energy to break the triple bonds of dinitrogen to form ammonium. And in the Haber-Bosch process, that energy comes from natural gas. So we use a lot of natural gas in the world to fix nitrogen and produce fertilizers. And so we produce these fertilizers to grow our crops for food, feed, fiber, and now even biofuel production. So if you remember what I said before, that we are feeding half of the world on synthetic fertilizers, what that really means is that we are feeding half of the world on natural gas, which is obviously not a sustainable approach. It's gas is a non-renewable resource. We are feeding the world on natural gas. So clearly there is a long-term sustainability issue. And if you have heard the news about the price of gas these days, it's increasing. And so that also drives the increase of the price of synthetic fertilizers, which for some countries is a significant issue. When you think that we use natural gas to make fertilizers to grow crops for biofuel, there I think it's almost ironic that we are using natural gas to make biofuel. And so obviously there it's not efficient. Besides these big picture concerns, there are also some very important ones, especially for developing countries where smallholder farmers, they really struggle basically for food production and trying to grow their crops in low nitrogen environments. The cost of fertilizer is limiting for them, and it's even worse these days with the increase of the price of gas. And even just the logistics of bringing synthetic fertilizers to poor growers is limiting. It's a significant issue. And both aspects of the cost and the logistics are very well showed in that book from Roger Thoreau, The Last Hunger Season. It takes place in Kenya, talking about corn growers in Kenya, small families trying to, with like less than 1 acre of land, trying to grow their corn under low nitrogen, struggling with the cost and the logistics of getting synthetic fertilizers. But of course, I'm sure that you also have heard about the environmental issues, the nitrate leaching and eutrophication of streams and rivers, degradation of coastal zones. In the US, there is that huge dead zone in the Gulf of Mexico, which is a direct consequence of the use of fertilizers here in the Midwest. So clearly there are these issues, and also global warming. Many of these fertilizers, when they degrade, give rise to greenhouse gases. And the production of fertilizers also contributes to production of greenhouse gases. So for all these issues, and for actually a long time, the community realized that we need to use more biological nitrogen fixation and try to rely a bit less on industrial fixation. So it's clearly not an utopic. There are papers on that topic for more than 100 years, but unfortunately, progress is slow. And so that's what I want to talk about today. What are the approaches to use more biological nitrogen fixation? So when you think about improving biological nitrogen fixation, there are some approaches that are more on the microbe side and some approaches that are more on the plant side. Obviously, to make it work, we need both of them. But some approaches focus more on one side than the other. The oldest approach that, if you look at the literature that people thought about, is to use legumes, the system in legumes, with the root nodules that host the rhizobia, and try to transfer that to cereals. There are papers from the beginning of the 20th century already trying to attempt that. It's a good idea. It's something that would be great if that works, but it's really long-term. My lab is working on a project towards that. You also have a project in the UK, the ENSA project, but it's really, really long-term. The other approach, which in fact is totally on the plant side, is why don't we take that enzyme, that nitrogenase complex, and put it into plants? Again, great idea, and several labs are working on that. We are trying a bit to work on that here, but it's a really, really long-term project. There are lots of issues to try to make the nitrogenase complex work in either mitochondria or plant chloroplasts. So these are exciting projects, but really long-term. And so today I'm going to talk about two approaches that are, in my opinion, much shorter term and that are complementary to these kind of big ideas that I just showed before. One of them is engineering the bacteria, taking bacteria that already associate with cereals and genetically modifying them to make them fix more nitrogen and release that nitrogen to the crops. That approach is entirely on the bacterial side. So kind of opposite to engineering the nitrogenase into plants, that one is 100% on the microbe side. And then there are approaches that are with the idea of exploring diversity, natural diversity. So people have tried to explore the bacterial diversity for decades, like taking nitrogen fixing bacteria in the soil, putting that on corn. It basically never really works. And I could tell you why I think it never works. But one aspect, critical aspect, is that very often the plant, the host, is limiting. We need better plants, better hosts to host these bacteria. And so that's why I was excited about exploring the plant diversity for such a trait like nitrogen fixation. And that's what I'm going to talk about. So first, I'm going to talk about an approach which is entirely based on the microbial side. And the idea here is basically, can we take nitrogen fixing bacteria that we call diazotrophs and modify them to make them fix more nitrogen, in particular when there is nitrogen in the soil, like when it's combined sometimes with fertilizers, and release that ammonium, fixed nitrogen in the form of ammonium to the plant. If we want to be able to use that in the field, because of regulation, we cannot use a transgenic approach. We cannot introduce any foreign fragment. So the game we play here is to do what we call cisgenics, which is to make modifications of using the genome of the bacterium and just moving parts within the same bacterium. We don't introduce any foreign fragment. And so the approach is typically targeting several aspects of the nitrogen regulation process. So the nitrogenase complex is encoded by NIF genes, and two very important regulators of NIF gene expression are NIF-A, which is a positive regulator, and NIF-L, which is a negative regulator of the NIF of the nitrogenase expression in presence in particular of nitrogen. When that nitrogen is fixed, then ammonium is produced, and that nitrogen is basically incorporated into glutamate to form glutamine via what we call GS-GOGAT complex, which is here. And many bacteria also have systems to recapture any ammonium that will have leaked out of the cell, so that they don't lose the ammonium that they just produced. So these are the typical targets that we focus on. I'm going to talk today about the work we did with John Peters in more detail in Azotobacter in particular. But I wanted to highlight that very similar work is done in collaboration with a company called Pivot Bio. Pivot Bio works on different microbes. So I have that NSF-funded project on Azotobacter, and I also work with Pivot Bio on different bacteria that are gamma proteobacteria, and in particular Klebsiella and Cosaconia. These are the main species that Pivot Bio is working on, always with the same approach of remodeling these bacteria to make them release more, fix more nitrogen in the presence of nitrogen in the environment and release ammonium. The problem with that approach is that these engineered bacteria often have reduced fitness. These bacteria fix a lot of nitrogen, release their ammonium, but of course they use a lot of energy for that, which is decreasing their fitness, and that's an issue. So that's why there is another approach, which is more, let's say, requires basically synthetic biology, which requires at this point really requires to have a transgenic approach, which is, can we use synthetic biology and more remodeling of these bacteria? And at this point, we need to introduce foreign fragments to control when fixation happens, and in particular when ammonium is excreted. And in that approach, what we do is that we take bacteria that don't fix nitrogen originally, and then we introduce the nitrogenase complex into these bacteria. The advantage of that approach is that we have better control. We can force these bacteria in particular to try to fix nitrogen only in the presence of a plant. Because with our approach currently with Pivot Bio in particular, of what I'm going to talk about, these bacteria fix nitrogen all the time. And so their fitness is strongly decreasing. If we can make these bacteria fix nitrogen only in presence of a plant, for instance, then we hope to improve their fitness. But the problem of that approach here, that second approach, is that it requires transgenic approaches at this point. And so we cannot, it's great for the lab, it's almost an intellectual exercise, but it needs to stay in the lab. All right, I'm going to talk in more detail about the project on Azotobacter vinellandii. Again, it's a collaboration with John Peters, and Florence Mus is the lead author on that. So Azotobacter vinellandii is a great nitrogen-fixing bacterium because it's one of the few bacteria that can fix nitrogen under ambient oxygen levels. So the nitrogenase complex is highly inhibited by oxygen, and in many associations where you have efficient nitrogen fixation, you need to protect the nitrogenase against oxygen. Well, Azotobacter vinellandii has its own oxygen protection mechanisms, and so it can fix even with 20% oxygen in the air, so with ambient oxygen level, which makes it a fantastic nitrogen fixer. Genetics of Azotobacter has been studied for a long time, and so we know quite a bit the mechanisms that control the expression of the nitrogenase. Again, I talked about NIF-A being the positive regulator, NIF-L being a negative regulator. So the idea we had for that project was, why don't we disrupt NIF-L and try to activate NIF-A? And so that's what we did initially with these two constructs, FM4 and FM5, where we inserted into NIF-L that kicks cassette, that disrupts NIF-L, and a promoter that activates NIF-A. Well, it worked, but it worked actually too well because these mutants where we disrupt NIFEL and activate NIFE, in fact, are lethal. And we even showed that they die just because they fix too much nitrogen. They fix so much nitrogen that they literally die from it. So we tried a slightly different approach by introducing that Kix cassette in reverse orientation. And in particular, we introduced it with and without an internal promoter. And interestingly, when we started introducing that disruption cassette, that Kix cassette in reverse, we got very nice ammonium excretion, a very nice ammonium excretion phenotype. So here it's that construct FM1, FM2, and FM3, where we disrupt the entire NIFEL. So FM1, we just disrupt the beginning, FM2, the end, and FM3, we basically disrupt the entire NIFEL. And all three constructs gave us, as you can see, after just even 24 hours, 4-5 millimolar ammonium in the medium. And after 48 hours, we exceed 10 millimolar ammonium in the medium, which is huge. So we were very excited to see that we had a very nice ammonium excretion phenotype. The next our next target, was that we realized that in all these constructs, we had a promoter in reverse orientation. And we started wondering if that gene that has been actually poorly characterized in Azotobacter on RNF1 was actually maybe important for that ammonium release phenotype. And so what we started to do is that we introduced not that whole Kix cassette, but just introduced endogenous promoters of Azotobacter in reverse orientation, where again, with these endogenous promoters, we disrupt NIFEL and activate RNF1 expression. And these constructs that are here basically show the same phenotype with 15, 16, 17 that are here, 15, 16, and 17 here. These constructs give us, again, a really good ammonium excretion phenotype. The advantage of using just these endogenous promoters is that then what we have, these bacteria we have are, again, cisgenics. That's the kind of bacteria that we can use in the field with the current regulations in the US. So that's what we show here, that we can get good ammonium excretion just by inserting a promoter in the reverse orientation, basically knocking out NIFEL and activating RNF1 expression. Interestingly, as you can see, not all the promoters are equal. We get the best ammonium excretion phenotype in the construct that are 15 and 16 that are here with these two promoters, and less ammonium excretion with SIG-B. And that actually surprised us, because when you look at the strength of these promoters, we actually see that SIG-B is a stronger promoter than the first two ones. And so what that means is that we really still have a lot to understand in the system, because it's not just to have the strongest promoter that gives us the best ammonium excretion phenotype. So my point here is that there are still, even if Azotobacter is a fantastic system that has been studied for decades, there is still a lot to know, even in Azotobacter, about how we can engineer, and many things to try to engineer these bacteria to fix more nitrogen and release ammonium. And that picture here is to show that not only they release ammonium in the medium, but when we put these engineered strains of Azotobacter in presence of rice, we were able to show that ammonium is transferred from the air, where, and for that technique we use 15N gas enrichment. So we put 15N gas in the air, and we were able to show that 15N gets into the plant. In that case, we used the rice plant. And so that fixed nitrogen is fixed by the bacteria, that nitrogen is fixed by the bacteria, engineered Azotobacter, and transferred to rice. So basically the system works. So the system works, great, but there are some issues. The first issue is decrease in fitness. Many of these bacteria that we engineer to fix more nitrogen and release ammonium into the plant grow poorly. And to the point where, for instance, with the first strains from Pivot Bio, these strains were great ammonium releasers in the lab, but when we put them in the field, they die in the field after 2 weeks. So obviously, they were too weak to be able to survive and compete in the rhizosphere. And so the game we are playing now is really to find the balance between the increased in all the strength that we generate between increased ammonium release and that decrease of fitness. what is that optimum that is mostly optimum for the field and not actually in the lab. And that's why I think that many people in that field of research failed to have practical applications for the research, is because that balance between fitness and ammonium release is very different between the field and the lab. And the second aspect is that different bacterial species will have that optimum at different levels. And so we, for instance, now use not only different engineered strains of one species, but we actually use combinations of engineered bacteria in the field to try to deliver nitrogen again from basically the planting time up to flowering time, which is the time where the plant needs the most nitrogen from the environment. So trying to find a good balance between fitness and ammonium release is one of our first challenges. And the second one is the fact that most of our crops prefer nitrate over ammonium. And our engineered nitrogen-fixing bacteria, they produce ammonium. And so how can we improve the transfer of that ammonium coming from the bacteria to the plant? I'm showing you here two kind of ideas that we are currently pursuing to try to solve these aspects, or at least to better understand the problem. The fitness, we try to understand how the microbial community in the rhizosphere of maize is going to affect the survival and the colonization of roots by our engineered bacteria. And for that, we use synthetic communities, small synthetic communities of the maize rhizosphere to try to understand how existing bacteria in the maize rhizosphere already sometimes may promote or prevent the colonization of the maize rhizosphere by our engineered diazotrophs. For the transfer of ammonium, we try to rely more on mycorrhizal fungi, which is one of my other interests in science. Is can we use fungi to help the transfer of ammonium from these engineered nitrogen-fixing bacteria to the plant? And really, our primary data really supports that mycorrhizal fungi are really efficient at capturing the ammonium from the diazotrophs and give it to the plant. So the idea here is that can we develop maybe even mixed inoculums between mycorrhizal fungi and engineered bacteria to deliver nitrogen to the crops. So summary for that aspect of my talk. First, engineering nitrogen-fixing bacteria. I think it's a promising approach. Its main advantage is that, well, doing bacterial genetics is fast, and so we can make progress quickly. It's not published yet, but our field experiments with Pivot Bio suggest that we can deliver about 10% of the nitrogen that maize needs through these engineered bacteria. Again, these are field experiments, so we need to conduct these field experiments over multiple years, but these are the data from our field experiments last year. So my guess is that we can probably deliver about 10% of nitrogen from the air by using these engineered bacteria for maize. The limits of the approach is that we need to work with cisgenics, meaning we cannot introduce foreign fragments if we want to be able to use these strains in the field in the US, and for the company Pivot Bio, for them to be able to sell these engineered bacteria, we need to use cisgenics. And that requires a lot of work. And so we need to try many combinations. And as I showed you, even for Azotobacter that has been studied for decades, there are still things that we are still learning about what makes a good ammonium excreter. And so it requires a lot of work and effort. And not only in the lab to design new bacteria, but also in the field to test these bacteria in the field. Evaluating nitrogen fixation in the field is long and requires multiple techniques. And so we need high throughput approaches, not only in the lab, but also in the field. And the last aspect of that approach, which is the main problem, I think, is acceptance of the technology. And I would say regulatory issues. We can do field experiments with cisgenic bacteria in the US, and these bacteria can be commercialized by companies in the US and Canada, but in many parts of the world, people don't want that. People are too concerned about putting genetically modified bacteria in the field. And I understand that can be why people are concerned. So the acceptance of that technology, I think, is limiting. So on one hand, it's a short-term solution that really works. I mean, to some extent, where 10% of nitrogen is significant. but at the same time, it's a technology that many parts of the world are not ready to accept. So because of that, I think we need to think about multiple approaches for different parts of the world to solve the problem. And one approach that would not involve any genetic engineering is, can we explore natural diversity? I'm a bit pessimistic about using natural diversity of bacteria because of history in my field that basically never really worked. And so I was more excited about exploring host natural diversity. Can we have better plants to host wild type, like natural nitrogen-fixing bacteria? And so that project basically was initially funded by a company. I need to say that I submitted that project to funding agencies in the US, and my project got rejected initially because it was too crazy. And the idea of going to explore plant natural diversity with zero data to support that it could work was not good enough for peer review. So a company, I was fortunate that initially a company, the Mars company, was willing to fund that. And very quickly, our project focused on maize coming from one village in the Sierra Mije in Oaxaca. And I guess that some of you have heard about the project already, where basically our project focused on the maize that is here, grown on the slope of the mountain. And the reason it's really attracted our attention is that it's maize that grows beautifully. People over there do continuous crop, like it's continuous maize for more than 10 years. Maize after maize, year after year, on a soil which is very low in nitrogen. They grow the maize in the traditional way with no synthetic fertilizers, and they have great yields. And so that's obviously attracted our attention. And that's what I'm going to talk about today, what we have learned from that. And I need to say that the environment is very humid. It's a tropical region with humidity over 80% and rain several times a week. So one feature of the maize grown in Totontepec is that first, the most striking one when you see it, it's a giant maize. It's 5 to 6 meters tall. It's basically twice the size of what people grew in the US. And the people that you see here in the field in Totontepec, they are not children, they are adults. And you can see the size of the maize. It's really the first thing you notice when you see it. Also, that maize grows over a really long season, at least compared to here, which is they plant in April, harvest in November. And so the maize requires 8 to 9 months to reach maturity, which obviously it's not something that can be, I mean, that means that maize cannot be used in many parts of the world. So it's great for there, but it's clearly a natural limitation of these accessions if you want to use them elsewhere. But really the fact that, that really attracted our attention initially is that they grow the maize in the traditional way, and it's continuous crop and no synthetic fertilizer. So obviously that was unusual. The data that really started to get us interested are coming from 15N natural abundance. And initially, it was a super high risk project. We only took one time point in 2010. And the technique we use is called 15N natural abundance. And it relies on the fact that in any soil, you have some 15N, and you have almost no 15N in the air. And so most plants take their nitrogen from the soil. And so most plants, their 15N content will basically reflect the 15N content of the soil. But when you have a plant that can fix nitrogen and get nitrogen from the air, the 15N content is going to decrease. And that's basically what we use as a way to evaluate how much nitrogen comes from the air in these plants. And right away in 2010, the data were suggesting that in the maize from Totontepec, we were seeing about half of the nitrogen coming from the air. So it was just one time point. So Pablo Zamora, who was the postdoc working on the project, came back in 2011 and 2012 and did more sampling during the course of the growing season. And what we see is that from the 3rd month after planting to the 6th month after planting, we see about, again, half of the nitrogen coming from the air. Before 2 months and after 6 months, we basically come back to 0. We don't see any fixation at all. 6 months, 7 months, that's the time when the plants start to tassel, to produce flowers, and we don't detect any nitrogen fixation after flowering time. So another feature besides the size and the long growing season of that maize is the fact that these plants are covered with roots, and these roots are called raised roots very often when they can reach the ground. But in that case, we prefer calling them aerial roots because they stay in the air, they never touch the ground. And these roots, when it rains, produce a lot of gel, what we call a mucilage. And here, oh, sorry, I have my classical movie. It doesn't work, sorry. I have to show that basically these roots are literally dripping with gel. Every single root here produces between 1.5 to 2 milliliters of gel. You have a similar gel production happening underground in the soil by underground roots, but it's in the range of microliters. Here we talk about milliliters. So it's really an abundant production of gel. So the next technique that we used after 15 and natural abundance was the acetylene reduction assay, what we call ARA, which is a way to measure nitrogenase activity, the activity of the enzyme. And so the advantage of that technique is that we can use it in different parts of the plant. We use leaf, stem, underground roots. These are real roots before rain and after rain. And as you can see, we detect basically nothing, a little bit in underground roots, but it's normal in any maize. But mostly a very significant activity when the roots carry the gel. Later in that paper, we also showed that just the gel itself, without any root, basically shows all the activity. So we show that the nitrogenase activity or the nitrogen-fixing bacteria fix nitrogen in the gel, and we don't detect anything in the root itself. So all the action happens on the root surface in the gel. So one problem that we have in the field of nitrogen fixation is the fact that we have many techniques, but all the techniques we have pitfalls and have issues. So I'm not going to go through all the evidence because everything was published, but basically for that paper, we used all the 5 techniques that we have in the field of nitrogen fixation to demonstrate that fixation happens in the gel and the plant basically is able to capture that nitrogen on the aerial roots. In terms of bacteria that do the work, we basically did metagenomics and isolation of bacteria and sequencing of bacteria. That picture here shows that the mucilage, that gel, that where nitrogen fixation happens, has a very different community composition compared to other parts of the plant. And here we show that it's enriched, that community is enriched with nitrogen-fixing bacteria. The composition of the gel, it's a polysaccharide, mostly of galactose, with a galactose backbone with some fucose, arabinose, and xylose residue. And we think that the gel itself is really a critical factor for nitrogen fixation, that the degradation of the gel provides the sugars for the nitrogen-fixing bacteria to have enough energy to fix nitrogen, but the viscosity of the gel provides, limits oxygen diffusion and provides a low oxygen environment for these bacteria to protect their nitrogenase. Another aspect of the trait is that the gel is also sitting on top of a root, and the root keeps pumping nitrogen out of the gel. So surprisingly, the gel has a very low concentration of ammonium and nitrate because the root is constantly pumping that nitrogen out of the gel. And so that basically makes the gel a really good environment for nitrogen-fixing bacteria to fix nitrogen. I want to show you that initially we worked again with the Mars project was entirely focused on the village of Totontepec, but later I started working when Mars stopped being interested in the project. Fortunately, I started working with the CIMMYT, and we sampled many more accessions throughout Oaxaca, and all these dots represent location of maize that the CIMMYT had in the collection, where we basically see the trait. And so we see more maize with the trait in the Sierra Mije in the mountains, but also over in the mountains of Oaxaca. But also we see it we find the trait in the lowlands of Oaxaca, in near the sea. And several colleagues, I don't have it on the map, but also showed me maize in Guatemala that also has the trait. I wanted to show you that the trait is actually coming from Teosinte, and not surprisingly, but as you may know, maize got domesticated from Teosinte parviglumis, but especially in the highlands of the mountains here, you have another Teosinte mexicana. And we find the trait not in parviglumis as far as frequencies at this point, but we find it in Teosinte mexicana. And yeah, you can see, here I'm just showing the example of the number of nodes with roots. These are the accessions from the Sierra and here that's the Teosinte mexicana. So at least part of the trait was basically in Teosinte mexicana. I want briefly to show you also that the trait is not unique to maize and Teosinte. We found exactly the same trait in sorghum. In fact, actually we found, I realized that the trait was also in sorghum, but it's like not even because in fact, colleagues in India in 1986 already made that observation that they actually put as a cover page of a conference book in 1986 that here they show sorghum plants with their aerial roots covered with gel. And at the time, they put that just as a cover page in that abstract book in 1986. They unfortunately have not published anything because they did not want to pursue that direction, but they already had evidence that they had fixation in the gel produced by the aerial root of sorghum. And so I started collaborating with colleagues at the ICRISAT in India, and we screened their sorghum accession and identified about 20 accessions of sorghum that have the exact same trait, where many nodes with roots producing gel after rain, and where we detect a good amount of fixation in sorghum too. These are preliminary data on sorghum accessions that basically carry many aerial roots or fewer aerial roots. And we see that these accessions in that case get, at least at this point of our data, show 10 to 14% of nitrogen coming from the air. So the accessions with more roots fix more nitrogen. So that's a project which is still ongoing for sorghum, but I think it's interesting from a practical standpoint. It's also quite interesting. The last data I want to show you here, and I apologize for being a bit late, is that we evaluated nitrogen fixation in hybrids between the Sierra accessions and what we call inbreds, classical commercial inbreds. We call them XPDPs. And so here in that technique, it's called 50N dilution. When you have high 50N in that technique, it means no fixation. Lower 50N, as you can see here from these accessions from the Sierra means that fixation is happening. And here, that first column represents the hybrids between Sierra plants and some classical inbreds. And what you can see is a segregation of the trait, that some individuals in the cross between Sierra accessions and inbreds, commercial inbreds, can basically show a good amount of nitrogen fixation. In other words, the trait can be transferred by classical breeding, by genetics. Initially, that project was funded by Mars. Mars, fortunately, stopped being interested in that project. They tried to patent it. Hopefully, unfortunately, it was declined, and they stopped funding that project. And so that basically, that allowed me to continue that project now with public funding from USDA, and it's a collaboration with the CIMMYT. And so now the work I'm doing is based on publicly available accession from the CIMMYT, and it's publicly funded, and all my work is public and publicly available. All right, what are we doing currently on that project? We try to determine the limits of the trait, meaning how much water is necessary, how does nitrogen in the soil affect the trait, is there a negative impact of the trait on the yield? That's what we're working on. Also working on genetics, we try to identify the genes using GWAS and QTL approach, and we're interested in the evolution of the trait. As I said, it's very likely coming from Teosinte mexicana, and can we demonstrate that there has been human selection by the native indigenous communities to select the trait, because that I think would be an important aspect of the story, to demonstrate that indigenous communities have selected for the trait. There is the breeding aspect. If we want to use the trait outside of the Sierra in a more diverse environments, we need to work with plants that are not that tall, that can grow in a shorter period of time. And so can we develop breeding? We started making many crosses, and we are very interested in seeing if people are interested in participatory breeding. Now that we know what the trait is, that we can evaluate, can we involve growers in that kind of breeding approach? We still study the microbial communities, and with the prospect of possibly developing inoculants that would maximize the trait. And the last aspect, which is more, maybe could be more controversial, but is there, could there be a synergy between the first project that I presented, meaning engineering diazotrophs, and that one? Can we think about taking bacteria that fix well in the gel produced by these maize or sorghum varieties and modify these bacteria so that they would fix even more nitrogen and release more ammonium to the plant? So there is potential for synergy. All right, I wanted to finish and acknowledge the funding agencies, NSF, USDA, and DOE. The first part of the project, as I said, of engineering bacteria is also a collaboration with Pivot Bio. And of course, I need to acknowledge the lab members and all the collaborators from different institutions. Thank you very much for your attention. I will be glad to take your questions.
Thank you very much for this wonderful talk, Jean-Michel. I'm sure there will be questions, maybe not many, but I think we can have one or two. No? Esperanza?
When you have overproducing bacteria, over ammonium excreting bacteria, will you were thinking about mycorrhiza taking the ammonium so it is not directly delivered to the plant, but there will be also bacteria in the rhizosphere that will take the ammonium as well.
Yeah.
And it will not reach the root.
Yes, yes. The idea of using, can we use nitrifying bacteria that would convert that ammonium into nitrate? The problem is, as you know, in agriculture, we often try to limit the activity and the presence of the nitrifying bacteria. That's something that we were thinking about. Can we engineer consortia between the engineered diazotroph and bacteria that would basically convert ammonium into nitrate, knowing that the plant would prefer nitrate? Yep, that's also a possibility. I'm not working on that, but that's something that we consider.
Fantastic work. Just a quick question. Can you give us the name of at least two of those nitrogen fixation bacteria which are in the gels of the serum?
Sure. Yeah, that's interesting. We find lots of Klebsiella and for the gamma protobacteria, Klebsiella, Enterobacter. We also find, as expected, Azospirillum, Herbaspirillum. Interestingly, lots of Herbaspirillum in Totontepec, and when we use the same accessions here in Wisconsin, not a lot of Herbaspirillum, but a lot of Azospirillum. So yeah, it's
So thank you very much, Jean-Michel.
Thank you.
We will move on. Thank you very much. Wonderful. So let me shortly introduce Esperanza Martinez. Let me shortly introduce Esperanza Martinez. She's investigator titular C here in this center. You all know her very well. She studied the career of Biomedica Basica, did her PhD with Rafael Palacios, a postdoc with Jean Denarier, and well, several sojourns in UC Davis in other countries as well. And I'm sure she will present us a wonderful talk. Go ahead.
Thank you, Otto, for the introduction. I will not present a historic overview. Rather, I will show you our interests on novel nitrogen-fixing symbiosis, both in animals and non-legumes, in maize particularly. Okay. So I will be talking about nitrogen fixation in the carmine cochineals, in tortoise, and in maize, as I said. So before the center was created, there were already reports in Nature and Science on termites fixing nitrogen. However, it took several years before the bacteria were properly described. And this is the paper by a Japanese group, Noda. Was my favorite one, and I always use it in my classes. And they show that novel genes for nitrogen fixation were found inside the termite guts, and there are spirochetes in the guts, and the genes that are expressed are from alternative nitrogenases. So later, a group in Israel led by Jurkevich found that there were nitrogen-fixing bacteria associated with fruit flies. So the reason why these insects fix nitrogen is because they have an imbalance of carbon and nitrogen. They have access to large amounts of carbon in the fruits or in the wood, and they compensate the diet, the poor nitrogen diet, with nitrogen fixation. And it is estimated, well, I don't know how, because those are very difficult estimates to be believed. They estimate that nitrogen fixation in insects may provide up to 30 kilograms per year per hectare. That is more or less in the level of some associative nitrogen fixation in non-legumes. So we asked the question if there were more animals fixing nitrogen, and we went to the safari in Tasco and collected feces from several animals. And we got nitrogen fixation in the feces of two animals. One was the African spolon tortoise. And from those feces, we could isolate nitrogen-fixing bacteria. And we decided not to work with the African tortoise, but rather with a Mexican tortoise that is called Copherus berlandieri, that is the Galapagos tamaulipecus. So from the Galapagos tamaulipecus, we got nitrogen fixation measured by acetylene reduction assay from all samples we tested. And from all of those, we isolated Klebsiella varicola, that is a nitrogen-fixing bacterium that we described in my group several years ago, associated in plants as endophyte. Klebsiella varicola from tortoise has a low genetic diversity, and we believe it is transferred from mother to offspring by the practice that many animals do, is that they eat feces. So eating feces is a way to perpetuate the bacteria in the same species. So Klebsiella varicola was described first in my lab as an endophyte, but in a collaboration with the Health Institute, we found that there were clinical isolates that corresponded to Klebsiella varicola. Nowadays, there has been an explosion of knowledge of Klebsiella varicola in different environments, and it is a minor fraction of clinical isolates. So we think that the Klebsiella varicola from tortoise has a limited genetic diversity because it is transferred, as I said, from mothers to offspring. So we could cultivate Klebsiella varicola easily in the lab, and we have the genomes from 2 strains, and the genomes show no virulence genes and very little antibiotic resistant genes. So they don't seem to be those environmental isolates that are circulating and that finally reach patients in clinical settings. But we did also experiments to directly test the virulence of several Klebsiella varicola isolates. And we found that they were not pathogenic. These are experiments that Monica Rosenbluth did in Galeria, and Viviana Romero made them in zebrafish, and in the Health Institute, they tested Klebsiella varicola in maize. In all cases, they proved not to be pathogenic. So we are searching for an animal model where we can test the effects of Klebsiella varicola in the growth of the hosts. And these are experiments that we will perform most probably in chickens. And the tortoises feed on cactus. And cactus are very good to make photosynthesis. They have high levels of carbon and low levels of nitrogen that favor nitrogen fixation. And there are other animals, these little insects, that also feed on cacti. And they spend their life attached to the clades, and we think that they fix nitrogen there. We chose these carmine cochineals because they have a particular interest in Mexico. They were used since pre-Hispanic times to produce pigments that are used for cosmetics or in food. And as I said, these carmine cochineals feed on sap. They attach to the cacti and stay all their life depending on the plant. So it was not unexpected to find bacteria from plants inside these carmine cochineals. And in the first analysis we made, we found Mesorhizobium, Raspirillum, and a beta protobacterium related to plant endophytes. For those of you who are in the field of nitrogen fixation, Aswarcus is not an unknown bacteria because has been the subject of study for many people working with rice or grass nitrogen fixation. So the novel beta protobacterium that we isolated, that we identified from the carmine cochineal, is related to Aswarcus, that is a plant endophyte. And we have worked for many years with the carmine cochineal, and we have been unable to grow in the lab the beta protobacterium. So it is becoming like an insect endosymbiont, rather dependent on the host for growth. Rafa Bustamante did some analysis of the transcripts inside the carmine cochineal and inside different tissues. And he found that Dactylopibacterium expresses NIF genes inside the carmine cochineals. It expresses NIF genes in the gut, in hemolymph, that is equivalent to our blood, and in ovaries. And the fact that many insect endosymbionts are in the ovaries is suggesting that they are transferred from mother to offspring in a more specialized way, because in humans it seems that we get it somehow, but here the insects that are metabolically and ecologically more adapted than mammals have a specific way to transfer their symbionts to the offspring, and that is amazing. So we have many genes differentially expressed in the different tissues, and nitrogen fixation genes are expressed indeed inside the cochineal. And furthermore, we can measure acetylene reduction in the tissues from the insect, not only the expression of the genes, but also the activity that is really indicating that there is nitrogen fixation in the insect. And we have explored now different species of Dactylopibacterium, not only the domesticated ones, but many different wild cochineals, and that they are very interesting because they are smaller than the cultivated one. So with all the information we have from this insect and from others that we are exploring also in the lab, we got like a hypothesis of how is the dynamics of bacteria in plants and animals. We published this review last year, where we also added our own results, and we proposed a cycle of bacteria between plants and animals. By eating plants, we eat the bacteria associated with plants. That sounds very, very obvious. We make a special point on endophytes, on bacteria that are inside tissues, because those endophytes would be protected from digestion and will reach the gut, the colon, where the bacteria digest the tissues that are covering the endophytes and will liberate the bacteria. When we eat bacteria and we have them in the gut, they will finish in the feces. And feces are a good source of inocula for plants. So we finally propose a cycle of endophytes and gut bacteria. And this has implications for human health, undoubtedly, because endophytes can be also pathogenic to people. So we don't want to eat pathogens as endophytes. And there's a whole story of how can we manipulate bacteria in plants to be good for human health. The last point I'm making is nitrogen fixation in cereals. Jean-Michel has made a wonderful explanation of why we have been interested in nitrogen fixation for decades. And it has been a long goal that we wanted to pursue. So why did we get involved in nitrogen fixation in cereals? Because we were working with Phaseolus vulgaris. And Phaseolus vulgaris in traditional agriculture doesn't grow alone. In many cases, it's grown in the milpa, and Luis Eguiarte already told us about milpa and the squash that is grown there. And in the milpa, maize and bean are grown together, and they share the same hole in the earth, and the roots are intermingled. So we wonder, do they share symbionts? And eureka, we found inside the maize the bacteria that form the nodules in beans. And then we started to be interested in maize associated with rhizobium. And indeed, we had some evidence that rhizobium could fix small amounts of nitrogen because we had the NIFH minus mutant, the wild rhizobium strain, and an overproducing symbiosis a strain that was HOP plus that reuses hydrogen and has higher amounts of nitrogen fixed. And we see difference in the growth of bean. And in the meantime, we were isolating lots of isotopes from cereals, and maize has lots of different bacterial endophytes. So as I said in the opening ceremony, I worked with Johanna Doverainen, and Johanna was a pioneer studying bacteria from cereals, and she described Azospirillum. And Azospirillum from different maize had the capacity to fix nitrogen. Later, many authors criticized her and said, nah, that's not true. It's mainly the auxin production that is responsible for the effects of Azospirillum in plants. But she kept working with many other diazotropes, and we used Azospirillum in a artificial consortium that we built with lots of different nitrogen fixers to help maize fix nitrogen in very artificial conditions. And we did a meta-transcriptomic study. And indeed, the gene that was the NIF gene that was expressed was from Azospirillum. And also we found expression of NIF genes from rhizobium, but in the acetylene reduction assays that are shown somewhere, we found that only Azospirillum was fixing nitrogen in the roots of maize detected by the acetylene reduction assay. So Azospirillum clearly fixes nitrogen in maize, and the fixation is higher when it has bacteria associated in the consortium. And we speculate that rhizobium, by making lots of gum, is helping Azospirillum fix nitrogen because it alleviates oxygen stress. and it makes like an oxygen protection. So Aspergillum is better to fixes nitrogen better in the presence of Rhizobium and other bacterium in this consortium. And the growth of the plants is indeed higher with the mixture of bacteria. So this is a common strategy now when testing inoculants, because we don't want to put all our hopes in one bacterium, but in a mixture of bacterium that could help to promote plant growth. And here we see finally the ethylene reduction activity. And Aspergillum fixes nitrogen in roots, in maize roots, a Klebsiella also, and a mixture of 5 different nitrogen fixers and Bacilla is better to fix nitrogen in maize roots. But the levels of fixation are not comparable to those obtained with Rhizobium and legumes. And the highest levels of nitrogen fixation in plants have been recorded in trees, in fast-growing trees in legumes. And those are also my favorite nitrogen fixing systems, because you can really evidence nitrogen fixation by the growth of plants. And these were obtained like 38 years ago, these photos, and 38 years ago, and the effects of nitrogen fixation is evident. And recently, like a few weeks ago, I took this photo in finger plants, and the same is found in mimosas, in acaciellas. We always see clear effects of nitrogen fixation. And in animals, we do not have a model like that, where you have inoculated tortoise and non-inoculated tortoise, and the non-inoculated is small like this, and the inoculated is big like that. No, we don't have that. And we don't have that in clams or in insects. So that's why I'm thinking to work with chicken, because maybe we can have small chicken and big chicken, that we can see the effects of inoculated plants. And what are we thinking to inoculate into chicken? The Klebsiella varicola from tortoises, because they have, we found that those bacteria do not excrete alanine when fixing nitrogen. And that's very interesting, because ammonium is toxic for animals. And so we don't want a high ammonium excreter, because maybe that can hurt the animal physiology. And if the Klebsiella from tortoises excrete alanine when fixing nitrogen, that could be interesting. And just remember that there was a report earlier that Rhizobium when fixing nitrogen in nodules could excrete alanine instead of ammonium. There has been a large controversy over that, because some people say, no, that's not true. And then Phil Poole also criticized the work of Emmerich. And finally, it seems that it is true that you can fix nitrogen and excrete alanine, because because the bacteria don't suppress all the assimilation of ammonium. And instead of excreting ammonium, it produces a more sophisticated product. So in summary, we found nitrogen fixation in carmine cochineals, a novel nitrogen fixing bacteria that we called Dactylopibacterium carminicum. Nitrogen fixation was detected in gut, hemolymph, and ovaries. In tortoises, we detected through metagenomic analysis and culture Klebsiella varicola. In cereals, we have diverse isotopes. Azospirillum fixes nitrogen, especially in a consortium. And so with that, I acknowledge all the people from my lab that have helped me through many years, especially my 3 technicians, Julio, Monica, Antonio. Thank you very much for your attention.
Thank you very much, Esperanza, for this interesting presentation. Are there questions?
I have one.
No. Well, Esperanza will be around.
Can I ask a question?
Sure. I didn't see.
Did you test your Klebsiella varicola on plants, on maize?
Yeah, we have tested Klebsiella varicola, but I must tell you something more interesting. When you published your paper, it had a big impact in Mexico. And then the CONACYT said, we need to support this type of research in Mexico, and we need to protect Oloton maize from patents. And then they made ¿cómo se dice? Una convocatoria. I don't know. CONACYT supports now this research, and they have given the project to INIFAP and to Rent, and you know the people there are working, invited me to work with them. So we now got Oloton seeds, and we have, and Pepe, Jose Luis Aguirre, will be the researcher working in this project. He's my PhD student that will get his degree in 2 weeks. And he will be the leader in the microbiology part of the project. And he now works with the maize eloton, the ones that you showed that have these aerial roots. And he isolated as an endophyte from the seeds without any inoculation, Klebsiella oxytoca. So that's a very interesting new result. And we are going to inoculate that new, instead of varicola that has clinical pathogenic related bacteria, oxytoca.
Okay, so I'm going to send you an email, because we isolated some Klebsiella oxytoca and some Klebsiella varicola that have no animal virulence change from the gel of maize and sorghum. And so we are currently working with that. So I will contact you.
Yes, we have to be in touch, because we were already in touch when we were proposing this project. Yeah. And we should collaborate on that.
Yeah, absolutely. Cool.
Thank you.
Okay, so thank you very much.
That was very nice.